my name is Victor Perrault and I'm a biotechnology student at the University of Aberdeen. Like many of you, my year started behind a computer because of a pangolin and his tiny virus friend, Sars-CoV-2, or COVID-19 for short. The virus spread quickly and already did lots of damages. Temporary measures such as quarantine and social distancing did not yet the expected results, and at the moment, a vaccine seems to be the only way out of this pandemic. And there is a good chance that we will go back to our favorite pub soon, as today hundreds of trials are on their way all over the world. From their development to their approval, each of them will follow a strict pathway to ensure they are safe and effective. After being tested on tissue culture or lab animals, trials start on a small number of healthy volunteers and are progressively extended to bigger and more diverse groups. Today, I will present you 3 different vaccines: Coronavac, created by the Chinese companies Sinovac and Dynavax, Moderna's mRNA 1273, and finally 8AD1222, resulting of the collaboration between Astra Zeneca and the University of Oxford. Sinovac is developing an inactivated virus vaccine, a method already used to prevent polio and influenza. This method is safe, but it needs boosters to be effective. COVID-19's virus is isolated from hospitalized patients, then replicated to produce big quantities of infectious material. Viruses are inactivated by the chemical beta-proprolactin, a substance that breaks the virus DNA so that it cannot reproduce or provoke any symptoms, but leaves intact the spike proteins. It is purified by size exclusion chromatography and depth filtration. As coronavirus spikes are polyimmunogenic, the adjuvant CpG1018, a toll-like receptor 9 agonist, is added to the vaccine to increase the antibody concentration, stimulate cytotoxic and helper cells, and generate a robust B-cell memory response. This vaccine has already been tested on numerous people. As a matter of fact, 90% of Sinovac staff tried it before all the safety assessments were done. China government gave an emergency approval for limited use in July on medical personnel and border workers. It was before phase 2 trial had even started. Indonesia wants to produce 350 million doses by the end of the year. However, at the moment, very little scientific data is available and most of it is not peer-reviewed. So the next challenge for Sinovac is to get an approval from other countries, as China methods are not very trusted abroad. To have a good understanding of the next vaccines, let's go back on the functioning of COVID-19. After entering the human body, the spike proteins on the membrane of the virus bind to a receptor called angiotensin-converting enzyme 2. The membrane of the virus fuses with the host cell and the infection begins. After extracting the genetic sequence coding for the spike protein from a sick patient, Moderna scientists designed a microRNA coding for the spike protein. However, by changing 2 key amino acids, they blocked the spike protein in its prefusion conformation. This sequence is encapsulated in fat droplets at a concentration of 0.5 mg per milliliter. Once injected, the patient cells use the artificial mRNA to produce spike protein that triggers a safe immune response from the host. This mRNA is self-amplifying. Moderna's first trial enrolled less people than Sinovac's, but it was more thorough as it assessed the impact of 5 different concentrations of vaccine. The trials also included elderly and people from different ethnicity to model American population. The vaccine production already started, and Moderna is working closely with the American government, but also with other countries such as Japan, Israel, and the European Commission, and the vaccine is eligible for a European market authorization. This vaccine has many advantages. It is cheap and easy to manufacture and induces both humoral and cellular immunity. However, Moderna has been highly criticized for not being transparent enough and many questions stay unanswered. What if the mRNA was not stable enough? Or if it were destroyed by the immune response? Will it produce enough spike proteins? Astra Zeneca was already working on a vaccine against a member of the coronavirus family, and they adapted their method to SARS-CoV-2. Here again, the spike protein are used as a tool to induce immunity, but this time, the full-length DNA responsible for it is inserted in a plasmid. The vector is a chimpanzee adenovirus, engineered to become harmless and lose its capacity to replicate. A leading tissue plasminogen activator is added to increase the vaccine immunogenicity. The resulting ChAd, OX1, and COVID-19 vaccine is inoculated to a patient. Once injected, the surface spike protein are produced and prime the immune system. The trials were mainly led in the United Kingdom and the United States, but an experiment is on its way in South Africa to make sure that the vaccine is safe for people living with AIDS. Other trials are planned in Russia and Japan, where the infection rate is higher, to confirm that the vaccine is successful. So far, the vaccine produced a balanced immune response and induced neutralizing antibodies as well as cellular immunity. However, it has mild side effects such as pain, chills, muscle ache, and malaise. No serious adverse events were reported though, and researchers proved that taking paracetamol would reduce the inconvenience. To accelerate the process, the European Medicine Agency started a rolling review, an emergency measure where data is analyzed before the trial is completed. Manufacturing already started all over the world, and Astra Zeneca promises a broad and affordable supply of the vaccine. Such vaccines are easy to produce and to store, cheap, and responsible for long-term immunity. However, in this case only 2 doses have been tested. Also, reaction to modified adenovirus is not fully known, and some fear that they might affect the sex cycle growth. Several countries already plan a strategy to vaccinate their population efficiently, but many questions stay unanswered. Will the vaccine price be fair? Will there be enough doses for everyone? And those interrogations need to be answered quickly, as vaccines are nearly here. Moderna hopes that the FDA will review his by the end of November. Oxford expects its vaccine to be ready by the end of the year. Sinovac thinks his will be available worldwide at the beginning of 2021, but their trial in Brazil just got stopped. However, other vaccines such as Pfizer may be ready before that. Let's hope that what scientists and the industry learn during this vaccine race will be useful next time a human will decide to eat a bat.